Hello again and welcome back to the lab and for our next discussion about blood groups. And in our last talk we were discussing antigens and antibodies. And antigens being those carbohydrates or lipids or whatever they may be on the surface of a red blood cell, and there may be lots of different kinds. Those things that are able to elicit or to provoke an immune response in the body. And when we say immune response, we mean the production of antibodies, which respond with these ends to those antigens. So if our group here was, say, group A, then we would call this antibody anti-A. And there are a range of different recognized blood groups. We have the A group, we have B, C, D, E, and there's even little c, little e, and a bunch of different ones, JK and Duffy and so on. They're all different kinds of chemicals that are found on the surface of the red cell, and all of which can potentially produce an antibody. And as we discussed, the antibodies are generally produced after first exposure to the antigen. So if you don't normally have the C group on your own red blood cells, then the body would recognize that as being foreign. If you ever received any of them through a transfusion, then your body would start to produce antibodies against that C group. It would start to produce anti-C. Now there is a special case in which this normal system tends to not apply, and that's what we're going to talk about mostly for this talk. And that applies for these two groups here, Group A and Group B. Now before we discuss why they are so special, we might talk a little bit about what exactly Group A and Group B are. And what we find is that your red cells will generally have a particular protein on the surface here called the H protein, or rather H. And that's almost like the building block for the Group A and the Group B. Now if you have the gene for Group A, then you will add a certain kind of carbohydrate onto the end of that H protein. And we will then call that the A group. Make that a little bit bigger. But if you have the H protein and you have the gene for the B group, then you would add a different kind of carbohydrate. You've got a little enzyme in your body that the gene codes for, and it would add a different kind of carbohydrate. So you might have either the A or the B or both or neither on your cells. So if we drew a little graph here to just display this, here is your group, what we would call the ABO blood group system. And so what we've got on our cells and what we've also got in our plasma, which is the fluid of your blood, which is where most of the antibodies are generally found. So we talk about, if you are Group A, then you would have the A version of that blood group on the surface of your cells. And as we said, we don't really know why, but this is a special case, the A and the B, because at some stage in your life, in your early childhood, you start to produce antibodies against either or both of these particular groups. It may be that there's something in the environment which is similar to these proteins, but for whatever reason, you'll produce an antibody against whatever you don't have. So if you're Group A, then you won't produce an antibody against A because your body recognizes that as yourself, but you'll produce an antibody against B. Sometimes we abbreviate it with just a little alpha sign, anti-B. And if you would then be straight away prepared to respond to the B group if it ever entered your system, even if you'd never been exposed to it before. Likewise, if your group is Group B, you have the gene that codes for this B version of the protein, then you will also produce at some stage in your early life antibodies against the A protein, so against what you don't have. Now if you're special enough to have both of these genes, and then you will have the enzymes that will be able to produce both of the blood groups. You can have a red cell that has both A and B on the surface of the red blood cells. And if that's the case, then your body will recognize both of them as yourself, and you won't make either of the antibodies you won't make any antibodies against either of those blood groups, because they're both recognized as self and not as foreign. And the fourth scenario is what we would call group O. And what that means is that you have the cell and you have the H protein, but you don't have either of the genes that code for A or B. So that will just remain as it is, and you won't have the A or the B on the surface of your cells. And if that's the case, then you will recognize both of these as foreign, and you will produce both anti-A and anti-B. You'll produce both the antibodies in your early childhood. So that's why it is a bit of a special case, because straight away your body has the potential to respond against the blood groups that it recognizes as foreign, even if you've never been exposed before. Normally in transfusion we have a little bit of a ability to give people blood without them responding to it, but not in the case of the ABO group. That's why it is important for us to match people's blood group right from the word go, because of this special situation. So when we hear blood groups, we don't just hear about A's and B's, we also hear about another antigen, which is this one here, which is we call the D group. Now let's just draw another little graph over here. When we talk about our ABO blood group, and there's different groups that we've listed here. We could be group A, we could be group B, we could be AB, or we could be O. And in addition to that, we also talk about this group here called D. All these antigens here, C, D, E, and little c, little e, not these two, they're all part of a group of a blood group system called the rhesus blood group. But this particular one, D, we might just call that RHD. It's the D version of the rhesus blood group. So if you have the D on the surface of your cells, it's not related to the H protein or the A and B, it's completely different. But if you have the D, then we might call you positive for the D. If you do not have the D, if you don't have the gene and therefore don't have that particular chemical on the surface of your red cells, then we would call you negative. So you would either be positive or you'll be negative for the D group. And that being the case, that's how we form all these different blood groups. When we talk about someone being A positive, then they are group A under this blood group system, and they are positive for the D group. If someone is B negative, then they're group B, and they're also negative for the D. So of course you could be B positive or you could be B negative, depending on whether you or not you have the D blood group. So that's the essence of our blood group system, and we could talk a little bit more about genetics and how you inherit these, and we might do that in another talk. But for the time being, this should be sufficient for us to work on, and in our next talk we'll discuss how we detect these particular functions and blood groups on the surface of red cells, and also how we can detect the antibodies that are present in somebody's plasma.